John Ojal, who will talk about the impact of pneumococcal conjugate vaccine in Nigeria, and then move over to Camelin, who will start from 5. So over to you, Ojal.
Thank you. I'm normally based at the Kemri Wellcome Trust program in Kilifi. And what I do on a day-to-day basis is to look at the value, or assess the value of vaccines in terms of impact, and extend this to look at costs, for instance. So today I'm presenting some work that we're doing in Nigeria. It's ongoing work, and I'll give you a flavor of what we found so far. Pneumococcus is an encapsulated bacteria, and the polysaccharide, the sugar coating in the bacteria, is the basis upon which it is serotyped. And we have about 100 serotypes known so far. They colonize the nasopharynx, and while they are there, they can stay for some days up to weeks, depending on your immunity, your age, and many other factors. They also compete to colonize your nasopharynx. So for instance, if you're already carrying one serotype, it's difficult for another to colonize you. So there is intra-species competition within pneumococcus. And they can clear from your nose with time. And these differences in how easy you acquire a particular serotype or how easy you clear it make these serotypes have different prevalences when you sample a population, because there is a fitness factor in how easy you acquire or you clear the bacteria. And while they're in the nasopharynx, they can invade your system. If they invade and end up in the bloodstream, they can cause bacteremia. If they invade and end up in your brain, you can get meningitis or just other forms of pneumonia or even otitis media if it invades your ear. And there are vaccines that have been produced to fight these particular infections. Unfortunately, at the moment, the vaccines do not protect you against all the 100 or so serotypes that are there. Actually, in many African countries, we do have the 13-valent vaccine, meaning it protects you against 13 of the 100 serotypes that exist of pneumococcus. And what has been observed is that if, for example, we introduce a PCV13 in Kenya, then those 13 serotypes go down in prevalence to some endemic or baseline level, but the non-vaccine serotypes, those that are not covered by the vaccine, shoot up such that your overall coverage prevalence of any pneumococcus remains the same. So they replace the niche that has been created, by the exclusion of, or the suppression of the vaccine serotypes. This map shows just a burden of pneumococcus as at 2015. The big dots on the map show the absolute estimated total deaths by this particular study by Brian et al. You can find it in the literature. And it is showing that Africa still has a very high burden, including Asia. And as at that time, and Nigeria, in terms of estimates in this particular year, had about 49,000 deaths estimated from pneumococcus. The second country that had the highest one in Africa was DRC. So many countries have introduced a vaccine, as you can see in the green color there. Almost every country in the world, but there are few, about 5 in Africa, that are yet to introduce any form of vaccine against pneumococcus. It's a very expensive vaccine, and most of the countries in Africa have introduced the vaccine with the help of Gavi, which is a global alliance for vaccine and immunization. And how they do it is, once you introduce the vaccine, you pay a small fraction of the vaccine cost, about 15% initially. So if the vaccine is $3, then you'll end up paying maybe $0.2 per dose, yeah? And over time, as your economy improves, you're expected to pay a higher fraction of the vaccine dose until you're fully self-financing that particular vaccine. And they have negotiated a cost with the manufacturers. So most countries that are funded by Gavi get it at roughly $3 per dose. But somewhere in the US, that vaccine is going for close to $100, so it's per dose. So it's quite expensive. And there are several countries that already reached this accelerated transition phase, including Nigeria, I'll add Kenya here, Gambia, and Djibouti, for example, in Africa. So there are many questions around how do we sustain such a very beneficial vaccine that is very costly. So we are looking at ways of cutting down costs and making sure that this vaccine is sustained. One way is to try get cheaper vaccines, and there is impetus in this. Like for instance, there's a Serum Institute of India vaccine that was pre-qualified recently that's going for $2 a dose, for instance, instead of 3. That's a huge cost cutting if you imagine birth cohorts in the millions in any population. We're also trying to see whether we can get more value for money by covering a wider number of serotypes in the population. Or can we, for example, remove 1 dose out of our schedule? Instead of giving 3 doses, can we give 2 doses, maybe in a 1+1 kind of schedule rather than 3+0, which most countries in Africa are using at the moment. So these are some of the questions that we are currently looking into. So, and Kenya, for instance, has already moved to the Serum Institute of India vaccine in its process of cutting cost. The other countries, obviously India, it's manufactured there, and Uzbekistan. And we've also done a study in Kilifi to look at does a fractional dose of PCV give you an inferior response? And we found that at a 40% dose of the current dose that is given in the regular program, the immunogenicity is non-inferior. So there's a possibility that, for example, we can use a 4-dose vial as a 10-dose vial. So I'll now focus on Nigeria after that particular background. When you think about your health, there are a few things you observe. You can observe when you're healthy and when you're diseased, but there's a lot in between that you don't get to observe. It's completely latent. And what's happening there for a disease like pneumococcus is you're meeting people. You might get infected when you meet them based on so many factors that I've mentioned, including just the risk of infection per contact, and all that. But once you acquire, then we also need to know what is the risk of getting disease when you have courage. That need to be factored in this latent phase. And if it's in a scenario where vaccination is already there, then we also need to understand what is the efficacy of the vaccine and how is it mediating all this latent process and making us observe what we're only able to observe, which is either courage or disease. And this highlighted area is where mathematical models will come in. So we come in with all the information I'm showing on the bottom right to characterize the latent stage so that we can have a data generating process that we relate to the things that we can observe. So we do need to collect some data, and we did this in 2 sites in Nigeria. In Kumbuso, it's a rural part somewhere in northern Nigeria, and Pakoto near the shores at the University of Lagos. And these were mostly courage studies. And Pakoto is rural. This is the population structure. So it's mostly compared to Kumbuso is rural. Compared to Pakoto, it has a younger population, and Pakoto has a higher population in the working age group. So it's more of a city or town kind of environment. And this figure shows the different studies that we've done and when we did them. PCV was introduced in Nigeria around 2016 in July for the rural area, Kumbutsu, and in October in the urban setup. We did several courage surveys in these particular sites. We did a vaccine coverage survey, 2 of them in the urban site, and 3 in the 2 of them in the rural site, 3 in the urban site. We also collected data on how people mix in these 2 populations. And what I'm showing here, the intensity of the color just shows the intensity of mixing. And in the right, left-hand side, you have the different age groups that we are looking at. We have 6 different age groups. And at the bottom, you have the age group, 6 age groups of the participant. And you can see the dark red part shows this a high intensity of mixing in Kumbutso among people aged, say, 15 and among people aged 15 to 20. And there's very little contact with the rest of the population with infants, for instance. And you can also see that the intensity of contacts in Kumbuso is higher than in the Pakoto, which is the urban area. So this just gives you a flavor of what you might expect in terms of transmission. But it's important data as we think about modeling. So our objective was to fit a dynamic model to this data we've collected, and then simulate some interventions that are important to the Nigerian NITAG. NITAG here means National Immunization Technical Advisory Group, because we're in communication with them. And then get some outputs of projections of the different scenarios that are of interest in Nigeria. And we use a model that is well presented by Yun Choi et al. So I'll fix your eyes on the top left part or panel of this graph. So we divide the population based on what they are carrying in their nasopharynx. So there are those who are carrying nothing. So we call them just susceptible individuals. You're susceptible to acquiring carriage of pneumococcus. There are those who are carrying a serotype that the vaccine can protect you from. So carriers of vaccine serotype, the state called V there. You can also carry a non-vaccine serotype. That's the other population in terms of carriage status. But sometimes you can carry both. By both here means a vaccine type and a non-vaccine type together in your nose. And there's a force of infection with vaccine, non-vaccine serotype, which is a red arrow. I don't know whether you can see my pointer. And then you can also, there's also a separate force of infection with vaccine serotype. And you can also clear these serotypes and move to the different state, which are the green arrows. So, for example, the green arrow from non-vaccine type to susceptible means you're clearing a non-vaccine serotypes from carriage or from your nasopharynx. Now, because there is vaccination, we assume that vaccination reduces your force of infection, but only the force of infection for the vaccine serotypes, the types that the vaccine can protect you against. So if you look at the second panel on the top right, you'll note that there's an extra function there, which is just the vaccine efficacy that reduces the force of infection. The vaccine in Nigeria is given as a 3+0 schedule, meaning you're given the first dose at 6, second dose at 10, and third dose at 14 weeks. And at each of those time points, you don't have the same level of protection. So the level of protection for someone who's given 1 dose is slightly lower than someone who's been given 2 doses, or someone who has the full protection from 3 doses. So we do have compartments that are also dependent on how many doses of the vaccine you have received. And the vaccine does not protect you for a very, very long time. So the vaccine protection also wanes, and someone who has 1 dose, for example, today, in a few years' time might move back to complete susceptibility. So if we imagine compartment A as those who are not protected at all by the vaccine, compartment B as those who are partially protected, and C as those who are fully protected, meaning they have had all the 3 doses, you can move between those bigger kind of sectors of the model depending on time. And we do estimate several parameters from this model. Remember I mentioned something around competition. To what extent does having a pre-existing carriage protect you from getting secondary infection? So that particular parameter we estimate from the model. So by what, to what extent does having pre-existing carriage reduce your rate of secondary infection? We also estimate the susceptibility to infection given a contact, because not all social contacts result in transmission. So we estimate the probability that a social contact will result in infection. And this we allow to be age dependent in 6 age groups. So those are 6 more parameters that we estimate. And the susceptibility to a vaccine serotype is also allowed to be different to susceptibility to a non-vaccine serotype. So we do have a ratio of those susceptibilities, which we also estimate from the model. There are a few other things that we needed to know that we take directly from literature because we didn't study them in Nigeria or fit using their data. And these include the vaccine efficacy, for instance, which we take from literature to be roughly 50%. This is against courage, vaccine efficacy against courage. And we also take the waning of the vaccine from literature, which is about 0.1 per year, roughly saying that the vaccine is really protect you for about 8 years. And we do have other external sources of data, like clearance rates. We get this from some observational studies in Kilifi, because we don't have such studies in Nigeria, how quick people clear the bacteria in their noses. And contact rates, I've said this we got from Nigeria, and routine coverage of the vaccine, we also got this from our fieldwork in Nigeria. So we do link those pieces of data to observation in these surveys using some likelihood approach. And we use a multinomial likelihood. So our X here is the data that you observe in the field in terms of courage in all these categories. So there's courage according to infection status, that is either vaccine, non-vaccine, or susceptible. There's also courage in the model according to your vaccine level or immunity, how many doses you've actually received, according to age group, according to location, because we have 2 locations, and the year of survey. So we put that in something called a log likelihood. And we also do have some prior information, which I mentioned in the table that we take, we assume for some of the parameters. And we take this through a process called Markov chain Monte Carlo. So it's a sampling process for estimating the unknown parameters. So what happens is basically you make a guess that we're estimating these 14 parameters, so you make a guess of the 14 parameters, then you put them in the model, run the model for 4 years within which you've done your survey, and then calculate the likelihood from those guessed parameters. So you set it aside, propose another set of parameters, yep. Then run it through the same process. So you have 2 likelihoods, yep. So you get their ratio, and you accept the proposal based on this particular ratio. I think this might be discussed better on Friday or tomorrow by TJ. Yeah, yes. But this is a process which you take through, you take the model through, and over time, then you have a chain of proposals of what those parameters will be that are accepted, and they should converge to values that match the data best. And this is showing how the model fit to the data. Each column in this plot is a different age group. So we have 6 age groups. So the first one is under 1, the second one is 1 to 5, 6 to 14, 15 to 20, 21, 49, and 50 plus. So the top row is for Kumbuso, and the bottom row is data from Pakoto. The points and spikes are the observed data from the courage surveys we did in these two locations over time. The year is on the x-axis, and the blue colors are for non-vaccine serotypes. That's the proportion of non-vaccine serotypes over time, and the red color is a proportion of vaccine serotypes over time. So we were satisfied to some level that the model reproduced what was observed in the field, to mean that the parameters that we estimated in that process mimic reality to some good level. So we could use the model to make some projections. So this is the projection we make in terms of the prevalence of vaccine serotypes over time in Nigeria. And the different colors, I don't know whether you can see properly there, reflect for different age groups. For example, in Kumbuso, which is the first panel, we project that the prevalence of vaccine serotypes will move from around 33-36% to just below 15% in about 10 years, for instance. And that's the same, the right side is a projection for Pakoto. And because the model is in the background actually estimating courage incidence, we can use the risk of having disease given courage incidence, which we've obtained from Kilifi, because in Kilifi we do have both courage and IPD surveillance, and we've been able to calculate that risk. So if we apply that risk to this courage data, the incidence data, not the prevalence one, we're able to estimate, convert these lines into incidence of disease. And the disease I'm talking about here is invasive pneumococcal disease. And look at, from the beginning, what the projection in terms of reduction in disease will be at each of the years I show here. The pink one is 2024, and the cyan color is 2030. So we're basically projecting that by 2024, which is now, there's been roughly a 50% reduction in invasive pneumococcal disease in Kumbuso, and about close to 60% in under ones, that is, in Pakoto. And you can see some herd immunity at play. There is a reduction even in disease, even in ages that are not vaccinated. And what if in 2028 Nigeria says this vaccine is expensive, let's give up? Because in January 2028 they're expected to fully fund that program of PCV. So this shows just a scenario where they stop vaccinating in January 2028, and in about 7 or so years, it's projected that they might be back to where they were before vaccination, roughly. So just a counterintuitive scenario. So we project that PCV has so far reduced IPD incidence in children by about 50% in those two locations. And the prevention of the remaining might need either extra suppression of the vaccine serotypes or perhaps a much wider vaccine covering more of the serotypes. We've looked at cost-cutting measures for Nigeria, which might include suppressing the vaccine serotypes by a series of catch-up campaigns, and then once a proper herd immunity effect is reached, or once circulation of vaccine serotypes is very low, they might be able to remove one dose from their program. So we've modeled that, but I'm not showing that today because it's not yet presented and approved by the Nigerian NITAG. So they have to see it first. So, but we've done that for them. And there are limitations to this work. And a major one is that there's no IPD surveillance in Nigeria, and we are using the risk of disease given courage from a different setting. And we know that it might not be accurate because this is closely host dependent. But what we've done is we've done a meta-analysis taking data from Malawi, Gambia, South Africa, Kenya, Mozambique, to just try and get a more representative estimate of this risk in the African population. And we might end up using that in the final analysis that we present for Nigeria. And we've collected data in 2 locations. We've tried to pick a rural one and an urban one, but we don't know how well all this might represent Nigeria in general. So there's still that a bit where we might not be too confident to say that this is what is happening in the rest of Nigeria. But we hope that maybe Kumbuso might represent all the rural areas in Nigeria, if it does, and Pakoto perhaps what's happening in the cities. So we, of course, plan to share all this and discuss with the Nigerian NITAG. Thank you.
Thanks, Ojal, for the great presentation, and also just showing us the impact of vaccines. And this greatly ties to what we learned today on the lecture on structured models and vaccination. I'd like to see if there are any questions for Ojal before we move to the next presentation. Any question? Yes.
Thank you very much for the great presentation. We enjoyed it, and I think it was robust enough. I wanted to find out a few things. Would I be correct to say this was a Bayesian week? And if so, I saw the propagation of priors into the modeling framework, which combined well with the multinomial likelihood. My question would be, the external information that was included that we can assume to be the prior information, did we do the sensitivity analysis to just see whether the estimates that we got were localized or they were robust enough not to influence the estimates that we actually got? I also wanted to know the prior distribution that was assigned to the prior information that actually we included in the model. Besides, my last point would be on the, I know that you used sampling-based technique, yeah, involving the MCMC, right? Great. I'm not sure which package you used. Was it the open bags, Ratwin bags? Was it 10 based on the Mtonian Mount Carlo? Or was it just another JAG which is just another Gibbs, which is other JAGs. Thank you. I think those are.
Thank you. Should I answer that
Let's
first?
take a
Okay.
few more then.
Thank
You
you.
had your hand up?
I had a brief question. Yes. Thanks, John. That was a fantastic presentation. Just had a couple of questions, but one naive question. You showed a picture of the population sizes in your two regions, and it seemed like the population size by age, and it was much lower in the 15 to 20-year-olds than it was in the other.
Population size by age, is it this plot? No?
I can't see the plot.
Oh, it's out here.
Yes, this one.
Yeah, okay.
Is that just because the rounding of the groups is different, or because it seems to increase by age and then drop off in the late teens?
Perhaps what you missed was this proportions of the population. Yeah.
What's the reason for that drop off?
Oh, so from 6 to 14 and 15 to 20.
Yeah. Is it just because they're different? There's a slightly wider age group in the 6 to 14s.
So the column for 6 to 14, that there's within Kumbutso, about 30% of the population is aged 6 to 14. That's what this is showing. But then only about 8% are aged 15 to 20.
Yeah, that seems like a big reduction.
Seems like a big drop.
Yeah, I was just wondering what's the reason
I can
for that.
double check their census data. That's what I extracted from their respective census information in those two locations.
Okay, wonderful.
Yeah,
Thanks.
yeah.
There's one question online, and Daniel asks if the team checked for cross-protection of the vaccine and the investigation against other strains of the disease. Maybe you can take those questions, and then we move to the next set.
Okay, I'll start with the first. Was this a Bayesian approach? I will say yes, to some extent. Yeah, because we do use prior information in estimating what the posterior distribution of the parameters we're estimating were. So it's a Bayesian approach, if you like, in estimation. Sensitivity analysis. Yes, we've done sensitivity analysis of the parameters. I didn't show them in this particular talk, but we've looked at sensitivity around our assumptions of waning, for instance, because we assumed 8 years. So we've looked at, what if it's 3 years according to some studies, or even longer, because some studies estimate about 20 years, so we do have that. We've looked at sensitivities, we've looked even at uncertainty in the contacts, because we do resample the contact metrics to try back in uncertainties around that and see what the impacts are. So we've done a lot of sensitivity analysis that I have not presented today. So the distributions used in the prior information, for instance, in the competition parameter, because we didn't want to, we don't have, it's really dependent on population. You cannot really use a prior, so I just use a vague uniform prior between 0 and 1, because that's where we know the competition should lie biologically in terms of whether that parameter makes sense. So for those that I'm saying here are fitted, we've used non-informative priors, okay? And for those that I list, for example, something like reduction in efficacy because you've not gotten the full vaccination. I'm using informative prior because we do have data to back that assumption up. But then I let the model still simulate and see whether there's additional information in the model beyond what the prior used was. But where I've used informative priors, I do have evidence of why I've chosen it to be that way. Package, whether I used JAGs or JAGs, no. I do write my own sampling or sampler, so I do have a function that uses some form of adaptive MCMC to code, though there are packages out there that you can use. But I do like coding my own because there are things that I'll want to save in the process that are very specific to something I want to use post calculation, so I prefer to code my own sometimes. Cross protection. So we've not allowed cross protection in this model. For example, we do not assume, because Nigeria introduced the 13 valent PCV, which has serotype 6B as one of the serotypes that it can protect you against. But we also know that if you're vaccinated against 6B, there's some level to which you can be protected from serotype 6A, which is not covered in the vaccine. But we have not included, for example, 6A as one of the vaccine serotypes in the model. So we have not included cross protection in the model.
Because of time, we'll move to the next presentation. And then if time allows, we can come back to the Q&A for both of them. Thank you.
Thank you.
We'll now have Dr. Kamelin present on malaria vaccines. And she's a postdoctoral scientist from Swiss TPH. So we'll let her take us through the presentation. Ojal, if you're able to stop sharing.
Yes. Okay. Okay, thanks. Can you see? I don't know. Oh, okay. I think it's now all right? Hi, everyone. My name is Cameline Orlando. I'm a postdoctoral scientist at Swiss TPH, and I work on modeling the impact of malaria interventions in malaria endemic countries. So the presentation that I'm going to share with you today is on some work that we did last year, if I'm not wrong, and it was used by the National Malaria Control Program in Mozambique to apply for the RTSS vaccine to Gavi. And that is the work that I'm going to share with you today. So I'll begin by giving a brief background on the context of the country and also the particular intervention that we're going to look at, which is the malaria vaccine. So as we all know, malaria is still a major cause of morbidity and mortality in a large number of sub-Saharan African countries. And Mozambique is one of these high-burden countries within the continent, with an estimated around 22,000 deaths annually and over 10 million cases per year. So the WHO supports a high-burden, high-impact approach to control malaria in high-burden countries such as Mozambique and Nigeria. And in 2023, the National Malaria Control Program made an application to Gavi to acquire or introduce the RTSS vaccine in the country. And this was the modeling work that we supported them to write in within that application to Gavi. So to support this NMCP's application for the vaccine, we performed an impact analysis of the benefits of deploying this vaccine in some of the districts in Nampula. So Nampula is one of the provinces within the country, and the whole of Zambezia region. Modeling, as we are aware during this course, and in also some of the seminars and talks that you've come across this week, can help us to estimate the impact of interventions and provide insights into choosing which particular intervention you could call optimal from an array of interventions that you may have. So malaria vaccines are some of the key additional interventions that have been recommended by the WHO to reduce the malaria burden in high burden countries. And the RTSS vaccine was recommended in October 2021 by the WHO in moderate and high transmission settings, which, as you are aware, Mozambique is one of them. So pilot studies in Ghana, Kenya, and Malawi showed that the vaccine had a partial efficacy against malaria in children. So this, the diagram on the right is showing sort of like the transmission pattern of malaria on how one human being, so that is here in the middle, can get infected by a mosquito, and this mosquito does introduce some parasites into the body through a blood meal, and these parasites within the body do grow in the liver, and one can end up developing malaria, both either asymptomatic, symptomatic, severe, and even death. And that is what that cycle on the left is showing. So I just wanted to highlight the particular pattern where this specific vaccine works within the body, and how you can, and how it blocks the transmission. So RTSS is a pre-erythrocytic vaccine, and it works within the liver stage. So once you get infected, that is at point A within the body, and these parasites migrate into the liver, and that is where this vaccine works. And it's through that it can block transmission within the body. So it targets the circumsporocytes proteins, and a portion of the hepatitis B surface antigen is acts to recognize this parasite early in its life cycle, because before it can cause malaria within an individual. So clinical trials that have been done in the countries that I've mentioned before, that is Ghana, Kenya, and Malawi, showed that the efficacy of this vaccine is around 36% in children under 2 years. Specifically, the clinical trials targeted children between 5 and 17 months old. So going directly to Mozambique, and within this context that I've talked about, Mozambique does, as in all other countries, it does not just focus on one malaria intervention. It does have a mix of intervention to reduce or control the malaria burden within the country. And the plans that we have there, so what we have on the left are showing the optimal, the intervention mixes within Mozambique. And these intervention mixes, we've given them some names in our modeling unit. So if you see something there called BAU, that means the business as usual, meaning everything remains the same. We are not introducing different intervention. We are not changing the intervention that we have. And in that business as usual, it means we have ITNs. These are mosquito nets in almost the whole country. So as you can see, the districts there, the stratification districts, and IRS in some of the regions. So that is the business as usual plan. And then we have a plan called CORE, which has ITN, IRS, and SMC in some regions and PMC in some regions. So PMC and SMC cannot be sort of like introduced in the same region. So as you can see, areas that have SMC in the plan don't have PMC, and the ones that have PMC don't have SMC. So these are the chemoprevention. These are the chemoprevention interventions that are done for malaria. Then we have the COPLAS. The COPLAS does have IT and IRS in more districts. It has SMC, PMC, and also RTSS. So this is the intervention that we are interested in. And then we also have IDEAL, which also has the 4 interventions. So zeroing down to the 2 provinces that I mentioned earlier, which introduced the vaccine within the country, that is Zambezi and Nampula. So Zambezi is the lower one. So these specific plans in the middle, that's the middle figure, it's zeroing down to those 2 provinces where we have Zambezi. So Zambezi is the lower one, which introduced the vaccine in the whole region, and Nampula is the upper one, which introduced the vaccine in some of the regions. So what we are looking at in this particular study is that we are evaluating the impact of the vaccine on top of the other. Pardon?
Yeah, is it possible to use the mouse and point to the areas? I don't know if it's possible. Is there a mouse?
I don't know if there's a pointer or a mouse. Okay, so I need to, because it's a Bluetooth, right? Ah, okay, yeah. Okay, and then where's the particular? They're giving me a mouse, I think. Ah, okay. So how do I use it? Is it on? Okay. And then, can you see?
The pointer won't work on the screen, but the mouse will.
So we need the mouse. Okay.
The pointer won't work on the screen.
Okay. Is that helpful or not? Can you see?
Do you see the pointer?
Is it that? No. Okay.
We have to minimize first.
Pointer, yeah.
Oh yeah, pointer options. Okay. 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 Thank you. Okay. Thanks. Okay, this is great now. At least we can be able to see the particular figure that I'm talking about as I move along with the presentation. So I was in this middle figure, which is focusing on the 2 provinces within the country. And the first figure that I showed earlier, that is the whole interventions that are done within the country for each specific plan that we were evaluating in this study. So as I mentioned earlier, we have Zambezi and Nampula. And Zambezi is the lower region, so if you look, for example, in I think PMC, the plan that has PMC, the one that is colored, that is the whole of Zambezia, while Nampula is the upper part, which we are introducing in some of the districts, RTSS. And what we are going to look at are, of course, these 2 plans that have the vaccines, that is the COPLAS and the IDEAL, and a counterfactual that has the other interventions within that particular plan, but without the vaccine. So the methodology that we use in our group to do country application for scenario analysis for malaria interventions modeling is what we do is that we use data to fit our model to past trends of malaria burden within a particular country. And this past trend can be the prevalence of malaria within a country or a region,
or the cases. These are the incidences. So we fit the model up to the last data point that we have the data, and from there we can be able to evaluate our various plans or scenarios. So that the plans are what we call scenarios. So we fit our data points to that particular data that we have. And at the moment for this particular study, we used prevalence in children between 2 and 10 years from MAP. And after fitting the model to the to the last data point that we have, and then from there we evaluated the various plans that we were interested in the study. So we used a model called Open Malaria. Open Malaria is a stochastic, individual-based simulation model which models the transmission of malaria in humans and also the vectors, which are the mosquitoes. So it has submodels. So it has a vector submodel, it has an intervention submodel, it has a case management submodel, it has a human host submodel, and all these models are linked within a simulator, and we fit our prevalence within the model calibration setup, and from there we are able to evaluate the impact using this scenario analysis. So the process in which we do this country application for answering a specific question, i.e. RTSS vaccine impact evaluation in Mozambique. One, we need to define the parameters that we require in the model. And these specific parameters include the biological parameters, intervention parameters, i.e. the RTSS efficacy, the geographic characteristics of the specific region, that is the Nampula and Zambezi, and we put that within the open malaria model that we have there in the middle and we've seen in the previous slide. And after fitting the model calibration to the data points, we then do evaluate our scenarios, do our analysis, and answer specific programmatic questions that we have been required to answer by the specific countries. For example, we were interested to evaluate the impact of introducing this vaccine in Mozambique. One could also be interested to evaluate the impact of extending the vaccine within the country. So within the model, these are the drivers of malaria transmission, as I mentioned earlier. So we have the vector, which is the vector parameters. These are the EIR, the biting behavior, the resting behavior, the feeding behavior, seasonality, and if there is any resistance within the country. As in Mozambique, there are some regions that have experienced resistance to some of the interventions used. And then we have the intervention parameters. For example, as I said, we are using vaccine on top of other interventions. So IEITN is sort of like a baseline intervention used within the country. So we also need those parameter-specific values for the interventions that we are evaluating in that particular plan. And then we have, of course, the health-seeking behavior. We have the health-seeking behavior for that particular region and other important parameters such as the importation of cases from other regions and countries. I think for Mozambique, that is unique, and also the population density. And after that, we will obtain the specific malaria outcome that we want in our study. Okay. The modeling assumptions is that we assume the specific trend that we are interested in, or the outcome is the prevalence or the incidences, which are the cases, and the drivers of transmissions I've talked about in the previous slide, and other contextual factors such as the population density and the specific strategy that the country is interested in. For in this particular case, they were interested in to understand the impact of introducing the RTSS vaccine. So these are some of the data sources that we used. So we used prevalence data from MAP, and this is an example of malaria prevalence in under 5s in Mozambique, and how these trends have been changing over time. And Nampula and Zambezi, as I mentioned earlier, I think they are on the where the pointer is. So I could say towards a bit northeast. As you can see from the map, those are the high burden areas within the country, and that was where this vaccine was being introduced. So these are some of the assumptions we used for the other interventions, such as ITN coverage of around 80% with a half-life of 2.5 years, ARS in some of the districts within the study area of a coverage of around 80% and a 5.4-month half-life, and a case management coverage of around 80%. These are some of the data sources for our interventions for IE, ITN. So there have been a lot of clinical trials and experimental heart trials that gave us the parameters for these particular parameter values. So I'll go to the results of the study. So we began by calibrating the model. And open malaria model is not spatially connected. So we have to fit each particular location, and by location I mean district. So in each particular district within Mozambique, we calibrated the model to the data. This is the map prevalence data that we had, and these are the the results of those calibration. So up there you can see something like model variance. So the open malaria modeling suite has a couple of modeling variants, and we were interested to see how these results were varying between the two model variants. And as you can see, so for example, the first plot that is for Zambezia and the Zambezia province, and a particular district called Alto Moloco, and in these two model variants, this is the first variant and this is the second variant, we can see that our model is fitting well to the data that we have. And so the data points are in red, I think red dots, and the calibration output are in the thick red lines for the one of the model variants, that is the R0063 variant, and in green is the R0068 variant, and with the confidence interval. So we saw that the model fitted well to the data that we had, the prevalence data that we had. These are also, I don't know if you're raising your hand. Oh, okay, that's all right. So these are also results for another region. This is in the some of the other districts in Zambezia, because this is a province that has a couple of districts. And then now going to our results that we are interested in, which are evaluating the impact of this vaccine and what is the impact of introducing it within that particular region of Mozambique. So we have the figure there on that is showing sort of like a line map of the outcome of the prevalence in under 2s. And on the left, we have Nampula, with gray being a plan without the vaccine, so that is the counterfactual. And in, I could say light gray, that is the plan with the vaccine. And we can see that introducing this vaccine under both the COPLAS and IDEAL does reduce the prevalence in under 2s, whereby in Nampula we have a reduction of about 10%, and in Zambezi we have a reduction of about 24% within our evaluation period, which is from 2023 to 2030. And then this is just a table going into details showing this impact of the vaccine, whereby we have a table with the reduction of prevalence, reduction in incidence, clinical cases averted, and expected deaths averted. So we have the province, those are the two provinces under study, and then we have the plans. So we have the COPLAS and IDEAL, the counterfactual, and the plan that has the vaccine. And as I mentioned earlier, in Nampula, under the Core Plus. Remember, the Core Plus had, I think, PMC in some regions, while I think Ideal had SMC, and it also had a couple of more districts with IRS. So of course we expect that it has a higher reduction in both prevalence, cases, and incidence. That's because of this additional impact of the other interventions. So I would like again to clarify that what we are looking at is the impact of the vaccine on top of the other interventions that are being used within the particular region. Here we also have a table showing the results of cases averted and the burden reduced in under 5s. And clearly we can see that in Nampula we have a reduction of about 10% under both plans. And in Zambezia we have a reduction of about, I could say 20%, because Zambezia, also remember, we are introducing vaccine in all the districts, as compared to Nampula, where we were introducing it in some of the districts. So in conclusion, to support this application for the NMCP to Gavi for the RTSS vaccine, our modeling study found that introducing the RTSS vaccine within the population will reduce the malaria burden when added on top of other interventions. I would like to acknowledge our other partners who we worked with within the study, of course the National Malaria Control Program, who came up with this particular question and the local data that we required. Then we had the CHAI team in Mozambique who supported us greatly also in the data and some of the analysis, and our group was the main modeling unit to come up with these results. Thank you, and I'm happy to take any questions or any comments from the presentation.
Thanks, Cameline, for the presentation. Before we take any questions, are you able to briefly mention the different interventions? As you mentioned, the acronyms like the SMC and the rest, if you are able to talk more about them for people who are not really conversant with malaria interventions.
Okay, thank you. I have one of our students here who is an expert in one of the interventions, so I'll rope him in to talk about some of the interventions. So I'll talk about ITN and IRS. So ITN, these are bed nets. I think we are all aware about bed nets, right? And we have used them and are maybe using them now. So these are bed nets that are distributed to, depending on the country strategy, like in Mozambique, these are given to, I could say, people who are vulnerable. So children, pregnant women, those are the ones who are targeted to be given these nets, and these nets are assumed to be used by the whole household. And then we have an intervention called IRS. This is the residual spraying of a chemical, pyrethroid, within households in a specific region. So that is IRS. And Roland, if you don't mind, because his PhD topic is on SMC and PMC, he can talk about that.
Okay, all right. Thank you so much. So SMC is seasonal malaria chemoprevention. So it's one of the interventions that have been recommended by WHO in seasonal area. So it's all about giving anti-malaria drugs to children, specific population under 5s. And PMC is a kind of SMC, but which targets other category of the population, which are under 2, I guess. So those are malaria interventions when we, the WHO recommended to implement them in seasonal malaria settings.
Thank you.
Yes, thank you for the presentation. If you could go to your last slide. Mine is just a comment. Second last slide.
It seems, okay. Is it in the under 5s?
The second last slide of your presentation.
Okay, the conclusion, okay.
Yes, so it's on the conclusion. I see that addition of this vaccine will lead to a reduction. This is how you've concluded. But I presume when we are modeling, we are estimating. So my comments just on the language, maybe it could have been better stated if you said the addition of this vaccine is projected to lead. So, I mean, I feel.
We had an argument about prediction, projection, forecasting since the start of the course.
Okay.
Because I don't know the degree of
Well
answer
noted.
currently you
That's a
have.
very good point. Yeah, because we are quantitative scientists, so that's a good point. Yeah. And maybe just to clarify, so in our group, we mostly present our results to non-modelers, like policymakers. And the structure of my presentation, as you have realized, maybe it's more geared towards non-policymakers who don't want a lot of language, like they want what I can say, a high-level presentation. So maybe I'm used to that language more because of the audience that I usually present to. But well noted, I agree with you very well. Yes, I think I was, yes, you had a question?
Yeah, I want to find out what drug is used in SMC intervention.
Roland?
I couldn't really remember the name of the specific name, but we can check quickly to be sure. Yeah, so it's SP. Okay, SPAQ.
Yeah. Okay, sulfadoxine pyrimethamine, SP.
It's okay? Why not?
Why not
She's
what?
asking, why not La? Why not La? Okay, so the history of antimalarials, the way they worked was that we all used to use SP, right? Before it got resistant. But when we introduced artemisinin La, let me just call it as La, the trials that were done, they showed that SP had higher efficacy in protecting against pregnant women and also seasonal malaria than lumefantrine artemether, and was less reactogenic as a drug, especially to pregnant women. No, reactogenic, less side effects.
Less reactogenic.
Yeah, to pregnant women. That's why it's been maintained as WHO 2.
Thank you. Yes.
Okay, thank you for that brilliant presentation. Yeah, I was just curious on the models, which you showed us there, the graphs.
The result or the model?
Yes, the graph, the various. It was on the calibration.
Okay.
Yes.
Ah, the model variants.
Yeah, exactly. Yeah.
Yeah. Okay. Good
So,
question. Yeah.
yes, I expected to see maybe a score distinguishing between which one was the best, yeah, or the fitted whatever, the fitted score. Maybe you could have talked about by a score of these, maybe used maybe likelihood or goodness of fit, so that we can distinguish which one was the best or how your model fitted. Eh? And then also I was curious to know how you validated your model, because you never talked on the same. Yes. Yes.
Point. Yes. Okay, we can take one more question and then I can take them together.
Okay. I appreciate your comment on the structure of your presentation and the audience that it targets. But I'm curious to know, you didn't really show us what your input parameters were and what you considered. In your modeling, did you consider all the doses of the vaccine, or did you consider if a child only got one vaccine? Because the malaria vaccine is given up to 4 doses. And I think if you're modeling from 2013, there's already data from pilot countries on the uptake. And the 4th vaccine uptake is a struggle. The coverage is a struggle. And how does that actually influence the reduction in malaria burden, coupled with the fact that the vaccine efficacy is actually relatively low compared to what we would expect for countries to adopt. How does that play out with the numbers? And then did you also factor in the waning efficacy of the vaccine? And how does that impact, and at what number of years do we actually see no benefit if there is no benefit, or do we continue seeing the benefits of the vaccine way after we can tell that it's not offering any other protection? And then I would like to know in what transmission settings the regions that you're talking about are in. Are they high transmission, moderate, are they seasonal? Was that a consideration when you're doing the model? Yeah, thanks.
Okay, thank you. So I'll take the two questions together, and I'll take any other afterwards. So I'll begin with the question on the model variants. I would like to clarify that the open malaria model is just one model, right? But it has subvariants, and these subvariants, the reason why we used the subvariants in this particular work was to test how robust our calibrations results were, and we picked model variants that had quite similar assumptions, so I could say they are quite similar to each other, to see how robust our calibration output is going to be. But going back to the open malaria variants, we do have these variants are, I guess, depending on your particular question, the specific question that you want to answer, right? So depending, maybe you're working maybe in a low prevalence setting, whereby maybe the like the malaria prevalence is almost dying out, or it's a bit low. We'll find that, remember, these are stochastic model, so you may find that we may have epidemic burnouts. So we may have some burnouts, and some model variants may be better for that particular setting as compared to the other. So maybe just to clarify, these two, we were not comparing two particular models. They are the same models, but model variants that had similar assumptions. So the difference between these two specific model variants are the proportion of the vector, like the biting patterns of the vectors that are found in that region. That was the difference between the two model variants, and hence why we didn't do sort of like a goodness of fit or a score, because that was not really our particular interest. We were just interested to find out on how robust our calibration outputs were. And then the lady there had a couple of good questions on the parameters, so I'll just go specifically to the parameters. So these are the parameters, like data that went into the model, i.e. the prevalence. This is map prevalence data and how it changed over the years, and how these were the data points that we were fitting into for each particular district. So remember, our model is not spatially connected. We have to fit each particular district, and these are the data points that went into it. So on the on the left we have the prevalence, and on the right we have ITN coverage, which also went into the data. We have data on seasonality, on the particular vector compositions, and sort of like their behavior, the biting patterns. And yes, I did not show those particular like values that went in, but I showed some of the data sources, such as the prevalence and the ITN values here on the spatial maps. So maybe what I can just add, why I chose maybe to show this rather than show the particular values, is because each particular district here within Mozambique has its own particular value. So each particular district has a different value for each specific parameter set. And then you had something, I think, on the vaccine, yes, I think so far both malaria vaccines, that is the RTSS, and the newly introduced R21 are, we could say they have suboptimal efficacy. And the assumption that we made in this study in terms of coverage, so we assume that the primary dose, that is 1, 2, 3, that is what I'm calling the primary dose, had a coverage of around 80%. And the secondary dose, which is now the 4th, has a coverage of around 60%. So that was the assumption we made in the study. And of course, it does the actual coverage. Though it's picking up, I think Ghana and Kenya, the coverage of the second dose is picking up, so which is good. But as you are aware that these results do depend on these specific assumptions that we have made on the efficacy and the coverage, and that is the context in which these results are true. And of course, they'll vary with different assumptions.
Thanks, Cameline and Ojal, for the great presentation. The speakers will be around for a few minutes in case there are any further discussions.
I'm also around tomorrow and Friday.
Yes, or any further discussions or questions you'd like to ask them. We'd like to end it there and thank the people who've joined us online for the seminars, which are part of the workshop on the mathematical modeling for vaccine-preventable diseases, which will continue for the rest of the week. And it's in collaboration with the Vaccine Impact Modeling Consortium and the Center for Epidemiological Modeling and Analysis. Thank you, and enjoy your evening for the online people. And for the ones in the course, we'll just continue with the schedule. Thank you.
Thank you, too, for having me.
So in terms of the schedule, we have again come to the end of the scheduled teaching sessions and practicals for the day. We will have supervision on demand for